A vacuole is a membrane-bound organelle found in the cytoplasm of various cells, most notably in plants, fungi, and some protists, animals, and bacteria. The term vacuole means empty space, but these compartments are far from empty, playing crucial roles in cell survival and function. The structure of a vacuole is dynamic and can vary greatly in size and shape depending on the cell's requirements. Key structural components include tonoplast or vacuolar membrane. This is the single membrane that encloses the vacuole, separating its contents from the surrounding cytoplasm. The tonoplast is composed of phospholipids and is embedded with various proteins, including transporters and channels like aquaporins that regulate the movement of ions, water, and other molecules into and out of the vacuole. This selective permeability is vital for maintaining cellular homeostasis. Cell sap, or vacuolar fluid, is the watery fluid filling the vacuole. Its composition differs significantly from the cytoplasm. The contents of the cell sap are diverse and vary depending on the cell type and its physiological state. Common components include water, the primary component, providing turgor pressure. Inorganic ions, salts, minerals such as potassium, sodium, calcium, magnesium, iron, aluminum, copper, and manganese. Organic molecules. Sugars, glucose, fructose, sucrose, and other carbohydrates. Amino acids, various amino acids, some of which are stored in high concentrations. Proteins. Storage proteins, particularly in seeds, and some degradative enzymes similar to lysosomal enzymes in animal cells. Acids. Organic acids like citric, oxalic, and tartaric acids, which contribute to the acidic pH of the vacuole, often between 4.5 and 5.0, but can range from 3 to 10. Pigments. Water-soluble pigments like anthocyanins, which are responsible for red, purple, and blue colors in flowers and fruits, and anthoxanthins, which range from ivory to deep yellow. Waste products and toxins. Metabolic byproducts, heavy metals, and defensive compounds like alkaloids and tannins are sequestered to prevent harm to the cytoplasm. Hormone metabolites. Certain plant hormones and their metabolic products can accumulate in vacuoles, playing a role in hormone homeostasis. Gases. In gas vacuoles, which are found in some prokaryotes, metabolic gases are stored. Vacuoles are broadly categorized based on their primary function and the type of cell in which they are found. Central vacuoles, also known as sap vacuoles, are most prominent in mature plant cells, often occupying 30 to 90% of the cell volume. Younger plant cells and animal cells may have several smaller sap vacuoles. The main role is to maintain turgor pressure against the cell wall, which provides structural support, rigidity to the plant, and drives cell expansion and growth. They store water, nutrients like sugars, amino acids, and ions, pigments, and waste products. They isolate and store substances that could be harmful to the cell. They contain hydrolytic enzymes that break down macromolecules similar to lysosomes. They actively transport protons to maintain an acidic internal environment, which is crucial for enzyme activity and ion transport, and help buffer cytoplasmic pH by sequestering excess hydrogen ions. Contractile vacuoles are primarily found in free-living protists, such as paramecium and amoeba, and some freshwater algae. They pump excess water out of the cell to prevent bursting in hypertonic environments. These vacuoles periodically swell, known as diastole, as they collect water and then contract, known as systole, to expel it. They may also remove metabolic waste products along with water. Food vacuoles, also known as digestive vacuoles, are formed in protozoan protists, some lower animals, and phagocytic cells, such as macrophages in higher animals. They are formed by the process of endocytosis, either phagocytosis or pinocytosis, engulfing food particles or external substances. They then fuse with lysosomes, which provide digestive enzymes to break down the engulfed material into smaller molecules that can be absorbed by the cytoplasm. Undigested waste is expelled from the cell through exocytosis. Gas vacuoles, also known as pseudovacuoles, are found exclusively in certain prokaryotes, particularly aquatic bacteria such as cyanobacteria and purple photosynthetic bacteria. They are not membrane-bound organelles, but rather aggregates of smaller gas vesicles enclosed by a protein membrane. While specific functions vary by type and cell, vacuoles generally perform several critical roles. Storage. Store water, nutrients like sugars, amino acids, ions, metabolic products, pigments, and waste materials. Turgor maintenance. Especially in plant cells, the central vacuole maintains turgor pressure, providing structural support and facilitating cell growth. Waste management and detoxification sequester harmful substances, toxins, and metabolic waste products, preventing them from interfering with cellular processes. Vacuoles contain hydrolytic enzymes for the breakdown of macromolecules, cellular debris, and even entire organelles through a process known as autophagy. Vacuoles regulate water balance within the cell, particularly in protists with contractile vacuoles. pH regulations help maintain cellular pH homeostasis by accumulating or releasing hydrogen ions. Defense in plants, storing defensive compounds like bitter-tasting chemicals to deter herbivores and pathogens. Cell growth. In plants, the expansion of the central vacuole contributes significantly to cell enlargement, allowing for rapid growth with minimal new cytoplasm synthesis. Transport and localization. Involved in the trafficking and localization of certain proteins within the cell.